the Genetic Alliance BioBank's vision is to revolutionize access to information and resources to enable the translation of research into services. That's a lot in one sentence, and we mean it. So in other words, we're not so interested in just providing a service that is a commodity. We're really much more interested in how do we do this in a way that actually moves the ball, so that we're moving and progressing toward better research for the various conditions that we all care about, and that those research projects, et cetera, turn into services as quickly as possible. So it integrates well into sort of Genetic Alliance's overall vision of how do people become the army that do these things. Definitely also integrates well with the cornet's vision, which is how do participants become the voice that moves translational science, especially comparative effectiveness and patient-centered outcomes. So our product is a biorepository, and again, as I said, registry solutions for advocacy organizations to stimulate research. Again, I'm not going to talk about the registry solutions. You can look in our webinar archive for webinars on peer, but I want to talk today exclusively about the biorepository software and system. And again, we're really looking at how do we stimulate research. What we've seen over the 20 years that we've been involved in this endeavor is that, in fact, research on the diseases that have biobanks, that have registries, definitely moves more quickly than on those that don't. And the best time to do such a biobank was 20 years ago. The second best time is now. So our process is a co-op model, allowing an extensible, interoperative, and cost-sharing model. So basically that means that if you don't see something in today's webinar and you need it, we get it. So it's a cooperative, just like in the old days, many of us belonged to, either shopping co-ops or farm co-ops or whatever. We do that so that we can afford something we can't normally afford. And when PXE International, my disease advocacy organization that I founded because my kids had that disease, 20 years ago when we started, we saw costs out the roof. They were $500,000 to bank 50 or 500 samples. We thought that was crazy, and so we built a model that's extensible, that's cost-sharing, that allows a very inexpensive biobanking compared to setting it all up on your own. So Genetic Alliance BioBank was founded officially in 2003. It was founded on the back of the PXE International Biobank, which was founded in 1995. So it was founded on the basis of about 8 years of experience, and so we pretty much knew what worked and what didn't work. We made a ton of mistakes during those years. My children will tell you, starting with, why is there a gallbladder next to the ice cream in the freezer? So we started in our house. We quickly, really very quickly, within days, moved to renting and trying to find resources that would take care of having a biobank, from a very grassroots, scrappy kind of system to then bringing it up to a point where we thought it could be cross-condition. And we made it cross-condition in 2003. At the time, there was a biobank board because it was a separate standalone entity. It's now part of Genetic Alliance, and it has an advisory committee made up of the people you see on the screen, some of whom have changed. This is for historical purposes. I wanted you to see where the individuals came from. Some of these people have now changed their affiliations because they're in different jobs, but still oversee some of the more ethical or large considerations. We did a landscape analysis and needs assessment in 1994, so that was the PXE International years. In 2000, as we began to think about how would we expand this for other diseases, 2006 and 2009, and actually I should have updated this. We did another one, a registry and biobank analysis in 2014. So we've done landscape analysis over the years to see what are the newer technologies, what's available, what are we missing, that sort of thing, and we continue to update things as we go. I'm not going to go through these screens, but they are screens to show you the kinds of things that we thought about when we did these landscape analysis. So what kinds of limb systems do people have? What sorts of registry kinds of integration? What sorts of general manipulation of samples and tracking? What kind of analysis? What kind of certification? what kinds of other sorts of processes can be done in-house as we analyze various biobanks that were available, and we continually looked at, should we continue to offer this? Is it still cost effective? Are we offering something that isn't available somewhere else? And continue to see that, yes, in fact, it does provide an offering for some of the groups that don't want to set this up on their own. So in this slide, we're simply showing a lot of the pieces that are available here, essentially starting with participation enrollment. The whole system is web-based these days. In the old days, we had to actually physically be in the biobank counting samples and sending spreadsheets off to each other. This is now done through a portal, an online secure portal that we offer. In addition to that, an IRB that covers all of this. So Genetic Alliance has its own IRB. We also offer boot camps and manuals, workbooks, wikis, webinars, so essentially all the kind of tools that a disease organization would need to set this up. And a lot of that results in things like informed consent templates, ability to look at what clinical information you want to track, sample collection, how to query the samples, data management, security, access and control, et cetera. So sort of a soup to nuts solution for advocacy organizations, whether they are just starting out or whether they're shifting to a new system like this one. So again, as I said, it's a cooperative model. So the advisory board representing organizational members, and that changes periodically as new members come on. We put more or different people on the advisory board, configures the best possible infrastructure. So we're revisiting that periodically, as I said, thinking about what do we need to update as solutions in science and technology improve. The members, which are the disease organizations, the advocacy organizations, bring passion, skills, and resources to the collective. So it might be that somebody in the collective learns to do one thing or another, or is figured out a way to attract more individuals to participate in their biobank, and they share that with each other. So there's no competition. Leveraging this collective resources, more services are possible. And then members also enable cross-disease research and maintain control of their own collection. So in other words, it might be that I need controls for some kind of discovery program, and I might take them from another condition. So PXE might ask psoriasis, can we borrow controls from you for XYZ? And of course, depending on tissue types and aliquot volumes and all that sort of stuff, we are able to do that or not. The disease organization maintains control of the collection. Genetic Alliance has nothing to say about where it goes, what happens to it. Genetic Alliance is simply providing the bank, the infrastructure, the maintenance and management. We are not managing the samples in the sense of deciding who in fact can use them. The members of the biobank receive training and mentoring by experts in registries and biobanking. So periodically, we provide a boot camp, sometimes virtually, sometimes physically. We also have webinars by experts in registries and biobanking as any of the services change or as we see new technologies arise. All of that. If the members themselves say, well, you know, we really wish we could learn about XYZ, then we get together the experts required and have that webinar done or that boot camp done. We also provide all of the templates. We probably have about 25 templates for any kind of documentation or protocols, including things like material transfer agreements, publication policies, all those sorts of things that allow individual organizations then to just sort of do a replace all. So we're really able to take care of individuals' needs and organizational needs that way. As I said, we have an IRB review of all documents and protocols, so all of the documents that a new member receives have been approved by the IRB, and then once they're customized for that particular condition, the IRB reviews them again, and you're given an IRB approval. IRB approvals are for 1 year, as is standard, and then the renewal comes up once a year as part of the biobank membership. We have a clinical data collection system with a customizable web interface for donor data entry, and that can be very minimalistic, or it can be as blown out as being also part of PEER or whatever other registry system that you're using. biological sample collection, processing, archiving, and distribution systems are all state-of-the-art. We right now are using a vendor called Precision Biosciences. Our vendor changes periodically, not because we think it's a good idea to keep moving around, but because we think that the vendors are either better or staying with the status quo, and we need to upgrade periodically, and we do so. We're very happy with Precision and think they have really done a spectacular job. We've been with them about 2.5 years. Web applications for data and sample management, and then the availability of multiple other genomic services. So again, either from vendors we're already contracting with, or we look for other vendors to take care of any needs of the members. And then mix and match modules. So an organization doesn't have to use everything. So the general biorepository services come from Precision Biosciences, sorry, I typed that wrong. Our cell lines are grown and cared for at Rutgers because we believe they are probably the best in the world at that. Phlebotomy services are offered by Exam One. So for example, if you have an annual meeting and you want phlebotomists at the meeting, they come out to your meeting wherever they are in the country. And then other services as needed. We have whole body harvests. We have specialized services for collection of unique things like plasma or urine. Those things can be taken care of as groups need them. So becoming a member is pretty easy. We've also tried to make this pretty easy because we want to have this for everything from the kitchen table organization all the way up to the more sophisticated ones. Potential members complete an application for membership designed to ensure organizational health and support when entering the co-op model. And the reason for that, if everyone can mute, I'm not sure if anybody's unmuted. The reason for that is that we want to make sure that the organization itself is ready to do this. We have had in the very early years a couple organizations that weren't able or didn't have enough infrastructure themselves. And I do truly mean that this can be done by a kitchen table organization, but it has to be one that's obviously committed to the long term and has some structures in place like scientific or professional advisors, that sort of thing. The application's reviewed and approved by the BioBank Advisory Board. And some of the requirements are completion of the application, organizational documentation. So for example, documentation of the 501c3 status, most recent 990, most recent auditor's report, copy of the DNO, and a voided check. Sometimes organizations do come to us who, for example, don't do an independent auditor's report every year because their income is not such that they would need it, or they don't file a 990 because they're under that bottom threshold, and we do work with those organizations as well. But we're really looking here for organizational health and the ability to continue. We like to look at the board of directors rosters. Again, early years we had a group join that we didn't look at in those days didn't look at the roster, and it turned out it was a husband and wife and their sister. When the organization began to falter, no one was reachable, and there were no essentially independent board members, making it pretty hard for the organization even to succeed, and they in fact had to default on continuing as an organization. We also look for a professional advisory board roster. Again, a really good thing to have in place if you're going to run a biobank, because you want to be able to get advice from others who are interested in research in the community. Not that we don't believe the board of directors itself and or even just the staff has the capacity. We all certainly do, but again, it's good to have that advice. And then we look for 2 letters of commitment and 2 letters of recommendation. Again, another way to tell us that, in fact, this is a viable organization and has interested members, interested advisors, that sort of thing. So once the application is approved, the member organization is then part of the Genetic Alliance BioBank, and then each organization obtains a Genetic Alliance BioBank IRB approval prior to collecting samples. And that's a very easy process. Again, because you're using templated documents that are already IRB approved, it's basically almost a replace all. There'll be a few things that will be different for each community, you know, for example, the age ranges that you're collecting, or the vulnerable populations, whether you are collecting from children or from pregnant women or from prisoners. Those sorts of things are things that the IRB will be looking for. And then the other big difference between us all that the IRB looks at is how are we communicating this to others? So what kinds of language are we using when we are communicating information to the community for them to join? Is the language coercive? Is it inviting? Is it appropriate? Is it pitched at the right level? That sort of stuff. So, so the IRB basically is quite helpful in helping the organization understand, and we obviously walk you through this too. How do you want to present this to your community? And again, same things I just said. We also provide things that aren't necessarily part of a normal biobank, like material transfer agreements with when you're sending a sample out from the bank, and you decide who to send to, when to send, that sort of thing. If you want to have a material transfer agreement, which we highly recommend with the researcher you're sending it to, what is the agreement between you and that person? Sample application form for researchers and other documents that might be useful to you. And again, they're all reviewed by the Biobank IRB. We also work with you to implement the procedures for clinical questionnaire. If you have one, again, many groups have a very simple one because they just want some phenotypic information with that sample. Others have more robust ones using whole registries, and that can be with us or not. And then also biologic sample collection. Do you want kits mailed to individuals' houses because your disease is sort of rare and you're not going to be doing, you know, wholesale collections, or do you want multiple kits sent to your annual meeting? Are you having some kind of you know, presentation at a professional conference, and you know that participants will show up there, or you're needing to collect controls, that sort of stuff. So we work with you on all of that sort of stuff. The next few slides, again, I'm not going to really go over in any kind of detail, but results of, for example, the PXE International Biobank have been immense. There's been an enormous number of papers published. There's been a whole lot of research done that's been spun off of having the collection in the biobank. Then to costs, which is always a great question for everybody. I should say when we started, the first 6 pioneer groups banded together, and we each contributed $25,000 each to start the biobank. In those days, that's what it cost. And for the subsequent years, we are actually able to drop that cost to about $12,000 a year. And we're pretty proud of that pretty low cost compared to lots of what we were hearing other groups pay, and even reading, you know, people quite excited about receiving a grant for $500,000 to collect 50 samples. We have since driven that price, I think, down to an extremely cost-effective level, where we're asking a $500 setup fee, a $500 IRB review fee, or you can use another IRB if you choose. I will tell you other IRBs are usually more expensive, and they haven't reviewed these documents ahead, and so there would be more heavy review. A monthly fee of $250 paid at the beginning of the month. That's to take care of all of the services that you receive and to keep the infrastructure going. And then services based on activity and storage fees based on volume. So in other words, if you have X number of tissue samples in -80, and you have, you know, X number of aliquots of DNA, also -80 perhaps, and then you have Guthrie cards, or you have other kinds of samples at room temperature, or you might have paraffin blocks, whatever they are, the storage fees are based on volume. And that volume essentially is, well, right now, the Genetic Alliance BioBank has 30,000 samples at -80. We negotiate with the biobank vendor, how many freezers is that? How many racks do we need in those freezers? What are those costs? Do we buy the freezers outright, which we have done sometimes? Do we rent space in the freezers? How do we want to figure that out that would be the most cost-effective and at the same time most reliable? What are the backup systems we need on those, and blah, blah, blah. And then we divide that cost across the biobank. They're pretty steady costs because we're able, at least so far over all these years, to incrementally build up as more volume increases, and so we have had no huge jumps in costs over that time. So I always like to end with this quote. It came in our early years that one of the participants in the biobank said, seldom is it that ordinary people like myself get to participate in these extraordinary events. I see donating blood in that case to the biobank as one of these events. Really getting that just contributing something as simple as blood, DNA, tissue, elective tissues from surgery, autopsy, body harvests at death are really a terrific way to contribute to science, and people really get that and they want to do it, as we've seen, especially, for example, in America and organ donors. We see the same thing in these communities who get that furthering the research in the community is going to be really critical, and that they can be a part of that. And that is the end of my formal presentation, under 30 minutes. I am happy to take questions. So again, if you could put those questions in the chat box, and I will answer them as they appear. So we have one question: will the PowerPoint be available for distribution? As I said at the beginning, the PowerPoint and the recording of this webinar are available on Genetic Alliance's website. Tanya, if you could switch to the slide with the URL, I think that's the middle slide. It is, yes, available at this URL, genetical.org/webinar archive in about a week or so, because it takes us a little time to process that. Another question: how many specimens have you collected in terms of number of patients, and how many have you distributed? So because that's managed by the individual groups, I can't give you an exact answer. I do know our volume right now is somewhere around 30,000 individuals banked in the biobank. And the number distributed, again, I have no idea, because I can tell you, for example, PXE International has probably distributed about 1,000 DNA samples, 4 or 5 whole body harvest tissues, lots and lots of paraffin blocks. But I cannot tell you what the other disease groups have done, because that is not my business. It really is the business of each of those disease groups. Approximate per sample fees. And we're happy to send the exact costs to you if you're interested. We don't just willy-nilly publish those, because for one thing, they do change annually, and so we don't want them out there and have them be old. But if you're seriously interested in the biobank and not just fishing, we're happy to send you those. You just have to send us an email. So approximate costs. Storage fees generally are somewhere around $7 a sample per, I think, you know what, I'm not even sure. I'm going to have to look those up. For something like sending a kit out, getting it back, and extracting DNA is $40. It's 10 cents for storing a paraffin block per month. So pretty low fees, again, as low as we could possibly get them, because of our ability to join together and get those fees down. Current monthly book of work in terms of patient samples distributed. Again, every single advocacy organization decides that on their own, and so I do not know. I can only report on the one that I take care of, which is PXE International. It really depends a lot on what kind of work I'm doing. You know, I'm about to do another whole exome screening of about 300 individuals with PXE, and so I'll send out 300 samples next month. But as far as what are other patient groups doing, I don't know. Next question: how do the nonprofits that are in the biobank interact with hospitals to get surgical and/or tumor tissue samples? Great question. So that's been actually like a work in process that I think has we've achieved quite a lot. And in the early years, that was pretty hard. It took a lot of heavy lifting on the advocacy organization side, and in a couple cases, for example, I even had to fly to the town where maybe an autopsy was being done or some elective surgery. Now we have put together protocols, and again, we share these with each other, so we're constantly improving them, that we give to one another to help the hospital understand what's happening. And so they include a whole host of things. They include, first and foremost, the IRB approvals, because hospitals need to see those. Although the individual having surgery actually has a choice to send their tissue anywhere they want without IRB approval, because it is being directed by the individual, we still make sure that that IRB approval is quite obvious to the hospital. And then we send the hospital an exact kit. So let's say it's a gallbladder, a mastectomy, you know, a liver resection, kidney, whatever, skin biopsies, we send the proper kit, and the kit will include exactly what it is that you want collected and how you want it collected. So for example, when PXE International does whole body harvest, I list 63 different sample types that I want. I list that I want some of them flash frozen, I want some of them in saline, and I want some of them in glutaraldehyde, which is a fixative. Then the kit that goes to the hospital has vials and boxes and dry ice and everything else all prepared for the hospital. So I certainly have had at least one conversation with the pathologist. We've worked out that they've received the kit, and that when the surgery proceeds, they will in fact make the proper correlation between the kit they've received and the samples. And then of course all the mailing labels and all that sort of stuff is given to them. In some cases it's much simpler. It's just one tissue sample or tumor, particularly in tumors. And again, we're looking at, I want a piece of it in paraffin, I want a piece of it flash frozen, I might want a piece in saline, and I'll send the proper kit. Obviously I don't send the kit. Our biobank sends the kit, and so I put the order in the portal. The biobank sends it out. If there's any question about it, because maybe it's very specific, or maybe I need a pathologist there to make sure that I'm getting exactly the right part of a tumor, then I have to contract separately with a pathologist at the hospital, and we'll again make sure all of that is well coordinated. Let's see, how much is tissue versus blood and serum is really, again, up to the disease advocacy organization. We don't make a determination about that. So, so that, again, is up to the disease group. Do you see this as something that would work for state newborn screening programs for storage of the residual newborn screening specimens? Yeah, absolutely, because I think in some ways that's a pretty easy storage play, especially since those are usually Guthrie cards and can be stored, for example, at room temperature. The biggest thing with them, of course, is indexing them so that they're available, and especially with the new requirements around consent, to be able to make sure that the right samples were stored in the right way and that they had the proper consent. And then also they could be retrieved if they needed to be, for example, if a family decided they no longer wanted it to be stored. So I do think that it certainly would work for that. How many disease groups are part of the consortium? Do we have a published list? I think on biobank.org there is a list. We just, in fact, today have talked about updating that website because we haven't updated it in quite some time. There are right now about 6 disease organizations. We have samples on 9 diseases. 3 of the early groups no longer continued as disease foundations, and so Genetic Alliance became the steward of the samples. We do keep a process whereby if a group is no longer able to be steward of the samples, then Genetic Alliance stewards that sample collection. So much information in a brief amount of time, yes. Do you assist groups in educating researchers about the existence of a biobank? Yes. So we do have quite a lot of information available, written in webinars. We create new ones when groups decide they need new ones. We have the biobank members themselves be the spokespersons for their banks and create webinars and materials. So we absolutely have a lot of material, a lot of information, and help the groups. As I said in the beginning, we're not terribly interested in just providing a service like you might find in a commercial biobank. We're way more interested in how can we leverage each other's abilities actually to help make this more viable. So it might be that I've not been able to reach out to a certain segment of my population, but I notice that another group is doing a good job. We have conversations with each other in a listserv. I might learn from inflammatory breast cancer that in fact they've been able to engage older people because they use XYZ materials, and then I just take those materials and modify them for my population. So, and then as far as educating the researchers themselves, we seem to have no problem with researchers being interested in these samples, especially young researchers, because well-characterized biological samples that are collected in a meaningful and careful way are not easy to come by, particularly that have longitudinal data associated with them. And so, again, we have bulletins we've written on this topic, brochures, webinars, and we share those with each other. So it's iteratively improving all the time by the biobank members themselves. To what extent do biotech industry companies use the biobank samples? So again, I can't answer that exactly because we don't track what happens for each of the disease groups independently, aside from sort of hearing from each other. And I do know that a number of companies have used the samples and have been very much delighted by them because they offer, again, this longitudinal look. They're well characterized. They're well cared for. They have good annotation. And so PXE International has definitely shared our samples with biotech companies that are in pursuit of treatments for us. I know a number of the other groups have done that as well. Another question of what % of the tissue bank is fresh frozen tissue versus paraffin. You know, I actually don't know. I'd have to go in and look at all the samples. And again, we're much less interested in those kinds of things because for your disease, you decide that. It's not that we would recommend that you have, you know, 50-50. That's absolutely not true for different conditions. You know, for example, in PXE, still lots of studies going on with regard to mineralization of various tissue. I need a lot of paraffin-embedded tissue. Some other group might need fresh frozen tissue because they're working with tumors. And so I don't like at all to make a comment on whether or not we should be at one percentage or another. How much time do we estimate is saved collecting and accessing samples? That's a great question. I'm not exactly sure how to answer that because I think because we have done this over and over and over, at least 30,000 times, let's say, we've gotten a lot of the kinks out of the system. We definitely had kinks in these now, you know, since 2003, so what is that, 12 years, we've definitely refined the system over and over and over, honed it to advocacy organizations. You know, again, this is not a biobank for a major institution. This is a biobank for advocacy organizations, nonprofits. We've really saved a ton of time, I think, in having the advocacy organization themselves figure out all these steps. And then in accessing samples, boy, that now is really great. You know, simply the researcher looks at either exported Excel spreadsheet or index of the samples and makes a decision about what he or she wants, or in some cases just simply comes to me and says, here's what I would like. I'd like individuals who are showing these phenotypic signs, and I'd like these kinds of samples on those individuals. You know, I might be studying retinal alterations, and so I want eyes, that sort of stuff. And I can have a sample sent out in 1 or 2 days compared to, I am sure, for that researcher to go find those from, you know, various collections around the world and or wait until those samples became available. I can't imagine the amount of time that we're saving those researchers. Can individuals elect to have tumor tissue from a prior surgery that's being stored in a hospital sent to the biobank? That is an excellent question also. Yes, absolutely. So depending on state laws, most states, individuals actually do have access to their own tumor tissue. There are some state laws that trump that, claiming that if the tissue's been excised, it actually belongs to the institution. I don't know of a single institution that has refused, and actually I'm going to say this and I'm going to contradict myself, that has refused to give an individual their own tissue. There have been a few. When they do that, they get such bad publicity, they don't ever want to do it again. And if anybody else hears about it, they encourage the hospital or the institution to readily give up the tissue. Sometimes they'll want to give up some of the tissue, particularly if you're still in active treatment. Sometimes they'll readily give up all of it if for example, someone's in remission. And yes, the individual can have a directive given to send that to your biobank. And again, we'd work with you to figure out, and so what does that look like? What kind of kit would be needed so that that sample arrives in excellent condition and is stored immediately at the new biobank? So some very simple things like we'd never have this done on a Thursday or a Friday. Although the biobank is open on the weekends, they're not accessioning tissue as much as they are during the beginning of the week, and so we'd look to have this sent out on a Monday, that sort of thing. And then again, making sure the right kit was sent so the sample could be transferred well. I've heard a number of groups state a limited few research requests for tissue. I understand how a researcher would wish tissue samples that include tumor cells. Do you make recommendations or discuss pros and cons of storing tissue samples with groups? So again, because I'm not an expert in this, what we would do and what we have done is convene groups of disease advocacy groups and experts. We've had as amazing people as Caroline Compton. We've had various people from other tissue banks, people from NIH, CDC, actually have these conversations with groups so that they had a better sense of what was the better thing to do. And then also, I also have heard of a number of advocacy organizations who seem to think that there aren't researchers interested in their samples. And I think this isn't a, if you build it, they will come. I guess I don't really believe that about anything. I don't think you can build technology and have people arrive. I don't think you can build a biobank and have people arrive. I think instead, we really do need to market these samples in a way that's appealing to researchers. We need to make sure we're targeting the right groups of researchers. We need to make sure we're articulating the value and articulating potential uses that a researcher might not think of. So I think especially for rare conditions, it is not always obvious to the research community what can be done. And I think lots of the things that have happened in recent years for rare diseases, for example, pointing out that they are exaggerations of pathways that then would be very elucidative for more common diseases that are using the same pathways are the way to sell this. But yes, this absolutely has to be sold. And I think the groups that are having limited requests probably are not doing as much marketing as they need to. And then one could say, well, you know, we're all kind of small, many of us, and we don't have the person power to do that. But I think, again, if we just repurpose each other's materials and methods of pitching, then I think we have a leg up against others that are maybe just waiting until researchers come. Do you also store clinical data associated with longitudinal sample collection? So the answer is yes. The biobank itself has a LIMS system, which is an indexing system that does allow some phenotypic information. It is not what I would recommend for a longitudinal data collection the way that I would recommend. And I would recommend that you use one of the registry systems that are out there. Genetic Alliance does offer one called Platform for Engaging Everyone Responsibly. Right now, our systems do not connect to one another. That's a project for 2015, so that the permissioning, which in peer is dynamic and granular consent for different kinds of data, will also extend to the sample collection. For now, those are two separate systems. The individuals consenting to the biobank probably also do, and at least in PXE's case, for example, give extensive clinical information, but that is deposited in peer, and the clinical samples are deposited in the biobank. Again, in 2015, as soon as I secure the right funding, we'll be integrating those two systems. Are protocols tailored in accordance with a targeted population, for example, diverse cultural, racial, and ethnic groups? Excellent question, and we definitely recommend that. In fact, the biobank members themselves talk a lot about how easy or hard it is in certain communities to collect, what kinds of materials need to be used, who should be the spokespersons in those communities, how helicopter science is probably not the right thing, and the community itself needs to be well integrated into the process and participants in the process. So whether or not a group decides to tailor its own protocol in accordance with a more diverse and better representative scenario isn't something that Genetic Alliance BioBank requires. We definitely recommend it, but we don't require it. So it really does depend on the group. I would say all the groups that are with us now are very concerned about those kinds of issues and are working hard to be as diverse as possible and to be as representative of communities, especially those that are generally underserved, albeit they have a lot of the same challenges that other communities do for registries as well as for biobanking in itself. Would it make sense to establish a biobank at the same time as a registry, or wait till after the registry has been established and is growing? You know, I think it's a good idea to do them at the same time, and here's why. I think you're capturing people's attention at a certain point. They're feeling like they're participating. They're feeling like they're giving something to you, something back, something for the future, something maybe for future generations. And it's not so hard at the same time to say either we also want to collect tumor tissue that you have banked, or if you're going to have surgery, we want to collect that, and or want to collect DNA from you. In the early days, all the DNA collection was done through purple top tubes and a blood kit that we sent to the person's home. Now, very often we're just using saliva kits. We can use either. We can even go to cheek swabs. We have all of those kits available. So I think at the very least, a saliva kit going out to a family that was participating in the registry is probably just another piece of their involvement, and they, in fact, might feel very involved in that case. So I think doing them together is fine. I could also see where you just wait and do it later, but I firmly believe what I said at the beginning. The best time to have established your biobank is 20 years ago. The second best time is today, and I think we often wait for things to be somewhat ideal, and this is the kind of thing you could start up pretty gradually. You saw the costs are pretty low, and even if you didn't want to go to high volumes right away, having some samples on hand, I think, starts you off, and you get comfortable with the process, which is pretty easy. PXE International did biobanking when it truly was an office in my playroom with only myself, full-time homeschooling my kids, doing daycare and running all the PXE stuff on the side. I had to learn a lot to do it and had a lot of great mentors, people like the people at NHGRI and NIH who helped me, but now we have so much of this kind of standardized that it is quite easy. Could I talk about the relationship with CINA and how the cornet pprn has assisted our banking efforts? So just to kind of put all those pieces together for people who might not be familiar, Genetic Alliance is one of the funded cornet entities, and so here's a whole lot of letters. cornet stands for the Patient-Centered Outcomes Research Network. That network was funded by PCORI, which is the Patient-Centered Outcomes Research Institute, part of the Affordable Care Act. And so CINA, which is the Community Engaged Network for All, is a pprn, which is a patient-powered research network, and as such, it receives funding from PCORnet. CINA and all the pprn have to pay attention to, but not necessarily bank. So we're all looking, all the pprn are looking at, do they want to biobank? Can they biobank? How reasonable is it to biobank? PCORI is not requiring that, and Genetic Alliance is coordinating now the pprn as part of the coordinating center for PCORnet, and we're certainly happy to offer this biobanking system to any of the pprn, and I do believe PCORI is fine with funds from the pprn being used to pay for the banking efforts. So how has this directly impacted the biobank? CINA groups, some of them biobank and some of them don't. They have biobanked, though, with prior relationship to Genetic Alliance Biobank, and some are looking at now coming into the biobank. So it's integrated. As we learn more with, for example, the biobanking task force that's part of PCORI or PCORnet, and patient engagement principles and all those sorts of things, we are definitely integrating all of those policies and principles into the biobank too. Do you encounter challenges with the transfer of biological samples from other institutions to the Genetic Alliance Biobank? So we definitely have encountered some challenges, but again, it is quite remarkable to me, and I have a very long history, personal engagement with this. how quickly institutions cooperate when they feel like it's patients, parents, participants who are driving the endeavor. So I would imagine it'd be really tough if I said I want Vanderbilt to send XYZ samples to Harvard and do a material transfer between two scientists. It's not impossible, and it's definitely way easier today than it was 20 years ago. But I find it very easy these years if it's me or if it's another advocacy organization asking for that transfer, and particularly if it's the individual themselves. So if someone says, you know, gee, you have XYZ tumor tissue from me, or you took my gallbladder, or I had a partial resection of liver or something, what I find is that that the institutions pretty readily transfer it. They are more concerned, more than sort of the kind of, in prior years, I think the idea of sharing wasn't great, and maybe even the intellectual property issues were more at the fore, and I find them more interested now in, does this comply with contemporary human research protections? Is the IRB in place for this transfer, that sort of thing? And as long as they see that that's there, there's no issue. I think also because Genetic Alliance has worked with so many IRBs, both Western IRB for our own peer platform, plus 10 or so major IRBs, Stanford and Yale, Harvard, Chicago, Vanderbilt, that there is a kind of precedent set, and we don't find any major issues. If a major issue was encountered, we definitely would take it on together. So in other words, if the disease advocacy group couldn't resolve it themselves, or wanted help even if they could, that would definitely be something that Genetic Alliance would jump into. And then we have a whole suite of advisors that are pro bono to us, working on things like our Free the Data project and other things. They're lawyers, they're ethicists, and they really love these kinds of problems and are very happy to help us pro bono solve them one-on-one. Our samples submitted with identifiers. So yes, they are submitted to the biobank with identifiers on them. That is part of the protocol that the IRB has approved, because we want to retain what's called chain of custody. The biobank, when it receives the sample, strips the identifier because it realiquots the sample and uses two code numbers. It uses a unique identifier that they assign that's part of their entire massive biobanking LIMS system of millions of samples, and they also use a code number that you've assigned the family. Different advocacy groups assign those in different ways. PXE and lots of the original groups assign a family number dash an individual number. So my particular number might be 0001-007. And that is going to also be on the sample. So there'll be two different identifiers on the sample, but they do arrive with the identifiers on them most of the time. I will say sometimes they don't. So for example, if I'm running a saliva collection at my meeting, so for example, I just had one in London at the House of Commons. I collected 60 saliva samples from people with PXE. I actually put their family code numbers on the files. I did not put their names or anything else on them, and sent them directly off to the biobank that way. And that's fine, because the biobank really is only interested in coordinating collecting with that number. If it was through a third party, or if I had a harder chain of custody to retain, then the name on the vial might be important as well, so that that association could be made at the biobank as well. But for the most part, the sample numbers that I've assigned are completely fine. They're unique identifiers. And again, this is all covered by the IRB. Have you done collections for mesothelioma or any for any patient or advocacy group? No, we have not. And are biobank samples available to be used for product and service development freely? Again, that is completely managed and determined by the advocacy organization themselves. So the advocacy organization would be the one that the scientists would engage with, or the company would engage with, to decide what would be done with that sample and what is within the purview of the researcher or the commercial company receiving the sample. And I think I might have come to the end of the questions. I'll give another minute here if anybody wants to type any other questions in. These were fabulous questions. The questions are always almost more useful than the presentation because you all can think of things that I might not, having been in these weeds for so long. Again, this webinar will be available in about a week, archived with both the presentation and the questions, so you'll be able to share this with whomever. I know that sometimes the other thing that we've done to help groups is we've done presentations for boards of directors, we've done presentations for research communities, so that you could make the right kinds of comparisons as you shop for solutions, because there are definitely other solutions out there these days, and we're really happy to do that as well. We are not selling the biobank at a crazy rate because we think people really do need to look at what they think works best for them. And again, these are sort of a la carte services. You can use one kind of service and not another. You can use DNA banking or not. You can just use cell line banking. You can decide you're just collecting plasma or urine or whatever else. It really is up to the disease group. We're not prescriptive about that. The one thing I would say is that I'm co-chair of the IOM Roundtable on Genomics and Health, and we in fact have just put together a short and formal study looking at what does it mean to collect DNA on people in research communities, and that it probably is more cost effective and more useful to have that DNA collected as you encounter each individual in a study or in a registry, for example, because later on it seems inevitable that many things are going to need to be parsed a la the kinds of precision medicine that we're seeing lately. In other words, which phenotypes with which modifier genes are going to be the most critical ones for treatments and therapies. You know, we've seen in cystic fibrosis, the drug Kalydeco treats 4% of the population, and you know, years ago, they may not have been able to do that subpopulation so neatly that they'd be able to have a therapy approved. So I think probably DNA is one of the things that most people will collect, but I think the rest of these kinds of samples definitely will be up to the various advocacy organizations and their needs. I think that's it. I don't see any more questions. I do want to thank everybody for participating. The questions were fabulous. And as I said before, if you have any questions or want more information, you just have to email me. I don't think we put my email address on the slide, but again, my name is Sharon Terry, and my email is sterry@geneticalliance.org. I think Tanya's about to type it. I'm very easy to find. If you just Google me, you'll find my email address everywhere probably. And so we, again, welcome any questions at all. We also can connect you with other organizations using the biobank to have a sense of what's happening for them as well. All right, thank